Hello student, for the 12th class. In this video we are going to study about the law of segregation and law of independent assortment. In the previous video we studied about the law of dominance. Now we are going to study about the next law is law of segregation. Student, this is also known as the universal law because there is no exception for the law of segregation. There is some exceptions are present with the law of dominance in the nature, but law of segregation is applied universally. There is no exception for the law of segregation. Law of segregation states that according to this law, each character is controlled by two factors, and these factors segregate from each other during gamete formation, such that gamete receive only one of the two factors and so that there is no intermixing of factors. Let's understand it. They said that each character is controlled by two factors. All of you know from the law of dominance, each character that also said that each character is controlled by two factors, and these factors segregate, or you can say separate from each other during the gamete formation. When we form the gamete, these two factors get separate from with each other, such that gamete receive only one of the two factors. One gamete can receive only one factor, and due to this, there is no intermixing of factors. Let's understand this by the cross. To understand the law of segregation, let's take the trait for the seed shape. So, what is the rule? Firstly, we have to write the parents. We also have to mention here male and female and male. So let's take one parent round seed, one parent phenotype round seed, and second parent phenotype wrinkled seed. So after the writing the phenotype, we will write the genotype, and all of you know parents select only the pure lines. So round seed homozygous parents should be there, and wrinkled small r. This is the dominant trait. This is the recessive. You can take make any trait cross here, but make sure you have to take the pure homozygous parents. One parent should be dominant and one parent should be recessive. Now after parent we have to write here gametes. And in gametes, so in the gametes formation, law of segregation is applied. According to law of segregation, each character is controlled by two factors. This character also controlled by the two factor, and these two factors get separate from each other during the gamete formation without any intermixing. अब ये दोनों factor हैं, they will be separate. Here for one parents, when gamete is formed, gamete is haploid, only one factor will be present. So you have to make the F1 generation. So in F1 generation, when gamete from this parent can fuse with this gamete, it will form RR. This is the result for the F1 generation. As you can see here that all the progenies of F1 generation are round. In this law, we have to just focus on the gamete formation, and we have to define the law of segregation here. Now come to the next law, that is the law of independent assortment. According to this law, two factors of each character assort or separate out independent to the other characters, factor of other character at the time of gamete formation, and get randomly rearranged in the offspring, producing both parental and new combination of the character. We can explain this law by using the dihybrid cross here. He said that two factor of each character assort or separate out independent to the factor of other character. If we select two character here, for example, if we take two character like round seed means shape of seed, and the yellow seed. Here I select the two trait, one for the shape of seed, second for the color of the seed. Now there are two characters. Now factor of these two characters separate out independently with each other. Like factor for the round seed, they segregate independently, and the factor of the yellow seed, they segregate. They comes to the offspring independently to the second character. There is no influencing between the two traits, two factors of more than one traits. Let's understand this cross by using the dihybrid rule. Let's understand how to perform dihybrid cross. All of you know, first of all, we have to select the parents. Same rule here. Then you have to write here parents. So let's take in the dihybrid cross are performed by using the two traits. Here we take the trait for the seed shape and seed color. So one parent showing both trait dominant form, and one parent show the both trait recessive form. So here we have to take both dominant round yellow seed. This parent will show wrinkled. And what is the recessive trait for the seed color? Green. Wrinkled green seed. Here you will write the genotype. Round yellow. Genotype is capital R, capital Y, capital Y. Wrinkled green. Small r, small y, small y. This is your parents. Next step is to make the gametes. Now first segregate about the round seed shape. These, all of you know, due to the law of segregation, these factors will get segregate with each other. So in one gamete, only one trait is one factor is present. Now segregation of this yellow means seed color factors also done. One y will come here and one y will come here. Same here. Firstly segregate the one trait factors small r. Now do segregation of another trait characters. These are the formation of gametes. Now F1 generation. This is the result for the F1 generation. Now this gamete can fuse with this gamete, it form this. And what is the phenotype for this trait is, look here, capital R, small r. This is the heterozygous state for the round or wrinkled. Which one express? Dominant one will express. So shape of seed is round. Look here, capital Y, small y. In trait for the color of seed, dominant one will express and dominant is yellow. So this is round yellow. This can fuse with this also, can fuse with this. Now then this will be the genotype for the F1 progeny and its phenotype will be round yellow. Same here with these two. These are the process for this gamete. This gamete can fuse with this can also fuse with this. So what are the result for the F1 generation is 100% are round yellow. But as you can see here, these F1 progeny are not homozygous, they are in the heterozygous state. Now you have to calculate or you have to form the F2 generation also. To calculate the F2 generation, we have to self-pollination between the F1. So F1 parents, these are the parents and its genotypes are, here you will write the parents. Their genotypes are capital R, small r, capital Y, small y, capital R, small r, capital Y, small y. Now focus here for in the formation of gametes for the F2 generation. Gametes. Now here 4 type of gametes will form. As you can see here, there is only one type of, in the homozygous state only one type of gametes are formed because both of these are the same. Here in this case also both of these gametes are the same. But for the heterozygous condition, here 4 type of gametes will form. Look here, focus here. This R can comes with this Y, then gamete will be capital R, capital I. There is chances that this R will come with this Y, small y. So capital R, small y. This small r can come with the capital Y. And there is probability that this small r can come with the small y also. Here, as you can see, 4 type of gametes are formed from the heterozygous parents. Same here, 4 type of gametes are formed. Capital R, capital Y, small r, capital Y, capital R, small y, small r, small y. All these are just order will get changed here, but on all 4 type of gametes are formed during the gamete formation of heterozygous parents. Now calculate the F2 generation. Here we will form a Punnett square to study the F2 generation progenies, because here 16 different type of progenies are going to form. Now for the making of Punnett square, you have to make a box. Something like this. Here we will write all the gametes for the one parent, and here we will write the gamete of the other parent. So let gamete of this parent will write here capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Here we will write the gametes of other parent. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Now here check both horizontal and vertical gametes. For this point, here gametes are capital R, capital Y, here capital R, capital Y. So the progeny genotype will be capital R, capital R, capital Y, capital Y. For this point, look both horizontal and vertical genotypes. Here capital R, small y is there, and here capital R, capital Y. So for the round shape of seed, capital R, and capital Y, capital Y. For this point, check both same horizontal and vertical. Here capital R is there, here small r, capital R, small r, capital Y, capital Y. For this point, capital R, small r, capital Y, here small y. Now same here. For this point, right, look at the both horizontal and vertical. Capital R, capital Y, small y. For this point, both horizontal and vertical. For this point here, R is capital R, small y, small y. For this point, capital R, small r, capital Y, small y. For this point, capital R, small y, small y. Same here. For this point, capital R, small r, capital Y, capital Y. Here small r and capital Y is there. For this point, look at the both horizontal and vertical gametes. Then you will get capital R, capital Y, small y, because small y is there. For here, small r is there, small r is there, small r, capital Y, capital Y. For here, small r, capital Y, small y. Capital Y is there and small y is there. For this point, right, capital R, capital Y, small y, because small r and small y is there. For this point, capital R, small y, small y. For this point, small r, capital Y, small y. For this point, small r, small y, small y is there. Now it's time to write the genotype for these progenies. All these are the F2 generation. This is the F2 generation is here. So capital R, dominant will express. Make sure we remember that dominant trait will express in the heterozygous condition. This is the homozygous condition. Round, capital R, capital Y, yellow. Round, yellow. Here, what will be the round and yellow? Dominant will express here. Recessive will express only in the homozygous state. Round, yellow. Here is heterozygous state is there, but dominant will express round. Capital Y is there, yellow, obviously yellow will form. Here is again both for both heterozygous condition is there, but dominant will express. In the shape of seed, round is dominant, and in case of color of seed, yellow is dominant. Round, yellow. Here, round, yellow, but capital R round, but small y and small y is there. So this is the homozygous condition for the recessive trait. So what is the color for the seed? Green, round and green. Here again round, yellow, round, green, round, yellow, round, yellow, capital. Heterozygous for the yellow. Capital Y, small y is there, but dominant will express. Round, yellow. Here small r, homozygous state for the recessive. Wrinkled. KL, wrinkled. Capital Y, yellow. Here wrinkled. Capital Y is there, yellow. For this, both are heterozygous, round, yellow. Here round and green, homozygous for the recessive. Here wrinkled, yellow, and this is both are the recessive homozygous, so it will be wrinkled, green. Let's find out the ratio for the four different genotype. Here you can see 4 different genotype are there: round yellow, round green, wrinkled yellow, and wrinkled green. Let's search the how many round yellow are there. 1, 2, 3, 4, 5, 6, 7, 8, and 9. Round yellow, 9 progenies are there. For the round green, 1, 2, and 3. For the wrinkled yellow, 1, 2, and 3. And wrinkled green will be only one wrinkled green is there. So this is the result for the F2 progenies. Ratio for the F2 generation is 9:3:3:1. This is for the dihybrid cross. This is showing the result of Love independent assortment, which state that each factor of two character gets segregated independently. Like round, we take the two parents, round yellow, wrinkled and green. But this round and yellow, they are not dependent on each other. They can transmit into the next generation independently. Like round के साथ आपका green भी आ सकता है, आपका yellow के साथ आपका wrinkled भी आ सकता है. Is it clear? So these are not independent. Round and yellow, they are not dependent on each other. So this is explaining your law of independent assortment. We will study about the some exceptions for these law in the next video. Till that, revise and learn these law of Mendel's.